It's my great pleasure to introduce Farhad Hormozdiari, who is defending his thesis today. Farhad joined my lab in 2010. He's been here 6 years. Unfortunately, he's leaving us to go to Harvard this summer. He has had a great career here. He published 37 papers as of today. He's been basically a staple in the lab. He even came in with a lot of research. He previously worked at Simon Fraser University with Sheikh Sinalf and at UW with Evan Eichler. He has made many contributions to sequencing, both before and while he was here. And since the time that he was here, he's transitioned into a first-rate geneticist and made contributions there that he'll be talking to us about. He also was recently just won the Best Dissertation Award in the Computer Science Department, which is a great honor. And so I'm very sad to have Farhad be leaving us, but I'm very happy for his great success.
Thank you, Eleazar, for the great introduction, and thanks for everyone for coming. I know it's very hard to come to defense at 10 a.m., especially in computer science department. So today I'm going to talk about some of the project I did during my PhD, and the title of my talk is Statistical Method to Understand the Genetic Architecture of Complex Trait. So as I'm trying to defend in the computer science department, I will try to give a very brief and simple introduction to genetics, especially human genetics. And we all know that all individuals carries their DNA, their genetic information is encoded in their DNA molecule, and the DNA molecule is a sequence of 3 billion base pair. And you can consider that each letter is consisted of ACTGs, and the combination of these ACTG will represent each individual uniquely. However, given any two individual, their DNA sequence is highly similar and only differ in 0.1% of the positions. And this differences is actually referred as genetic variation, and they account for the differences that you see in the trait, like for example, the difference that you see in the eyes or height between the individuals, and they play an important role in the disease. Now, genetic variation come in different flavors, and the most common one is called single nuclear polymorphism, or SNP, which is a single mutation between two individuals. And here I'm showing an example between these two individuals that in this position they differ. One individual have A, the other individual have T. And there are other kind of genetic variants, and they are in a larger scale, and usually they are referred as structure variation. But during my talk, I will emphasize on SNP, and all the method I'm explaining today can be applied to them as well, but for simplicity, I will just use the concept of SNP. So in genetic study, one goal is to find this genetic variation, and the other goal is to try to figure out which of these variants are affecting the trait or the disease. And a simple process that people do is that they collect individuals for one of the phenotype that they're interested. For example, in this toy example, we are considering the cholesterol level, and we collect all the individual, and we want to see if any of the SNPs is associated with this phenotype. And what we do is that we are looking for a pattern. So for example, in the SNP1, you can see that individuals who have A or T have no specific pattern to people who have high or low cholesterol. So this SNP most probably is not causing this phenotype. However, if you look at the second SNP, you can see all the individuals who have C in this position have high cholesterol. So it's possible that this SNP can be a potential causal SNP for increasing the cholesterol, or for some individual decreasing it when you have the G allele. So how we can figure out these SNPs? There's a simple statistical test that people perform, and there are two hypotheses. The first hypothesis is that this phenotype is independent of the SNP, so it means that there is no correlation between the phenotype and the SNP, and they assume that this simple model, that the vector of phenotype, which is represented by y, is equal to this very simple model mu plus epsilon. And the mu is the phenotypic mean, which is a constant number, and E is actually the environment noise and measurement, the measurement and the effect of environment. And how you can represent in a graph, in the x-axis is the SNP, the allele that you have, and the y-axis is the phenotype value, and each point represents one individual. And the best line that you can fit maybe in this data is this horizontal line that represents this model, which means that the SNP has no effect towards the phenotype. The second model that people test is actually fitting this model, which is to present that alternative hypothesis. And you assume that the phenotype is actually equal to mu plus x beta plus an error noise, and beta is the effect of that SNP. And what you're testing if this beta, which is the slope of this line, is actually significantly non-zero. Now, if we can perform this test, this is testing one SNP, we can perform this genome-wide on all the SNPs, and the output of this will be a Manhattan plot, which is shown here, which is the x-axis is the variant position, and the y-axis is the significance score. And the higher the significance score, the higher the correlation or association of that variance to the trait. With this introduction, I will get to the papers that I published during my PhD, and I can categorize into the four sections. And the first one actually was the sequencing, and we all know that there are these machines that revolutionized the field of genomics, that they can sequence short segments of DNA in a very time and efficient manner. But this short segment and this machine raised a lot of computational problems that we tried to tackle during my PhD. The second category of the project are related to genome-wide association method, how to increase power or how to perform phenotype imputation or summary statistical imputation, which I will talk about one of the projects from these categories. And the third category, which is trying to get the GWAS result and trying to find the causal variants or causal gene, and most of my talk today will be emphasized on this category. And the last category is genome privacy. It means that can we share our DNA information in a way that people can perform some specific function to figure out some limited information, but the privacy of the individual who shared the information is preserved. So today, mainly, I will only focus on these three projects. I will try to explain them in a higher depth compared to the other project. I will explain two of the projects in a very high level towards the end of my talk. So the overview of my talk is as follows. I will first explain one of the methods that we developed called CAVIAR, which can perform fine mapping in the variant level. And then in the second part of my talk, I will extend this method and we propose a new method called CAVIAR gene, which can perform fine mapping on the gene level. And in the end, on the third part of the fine mapping, I will explain one of the applications of the CAVIAR model. And the last project I will talk about is about phenotype imputation. So as I mentioned in my beginning of the slide, I said GWAS was extremely successful, and it found thousands of variants that was associated with different trait and disease. But the question now people are interested to ask is that which of these variants are actually causal? And the causal variance is variance that is responsible for peak of association. So for example, when we see this peak, we want to know which variance actually is responsible for it. And the process that does that is usually referred as fine mapping. So you may consider that this problem seems like a very easy problem. Just pick a significant threshold and consider anything above it to be causal. So here I'm just zooming in one of the region, and you can see we picked this significant threshold. However, there are too many variants that pass this significant threshold, so it's not obvious which of these variants are actually causal. And the reason this can happen is possible that one of these variants are actually causal, and other variants are in LD with this variance, and they seem to be significant. So how can we solve this problem? Let's go over a very simple example. Here I'm showing a region which has 35 SNPs, and the x-axis is the position, and the y-axis is the significance score. And the gray triangle here is the LD. And each diamond in this triangle represents the pairwise correlation between the two SNPs. And the darker the diamond, the higher the correlation, and the lighter the diamond, the lower the correlation. Now, if I give this region and ask you which SNP is causal, based on your intuition, you can figure out that this SNP is the causal SNP. Based on the LD and how the statistic was distributed, this is actually a very reasonable answer, and this is the right answer. And how we know it is that as we implanted this variant to be causal. So you can assume that using your intuition, you can be able to answer this question that which variant is causal. However, not all the cases are that simple. Some regions are more complicated. Here I'm showing a very complicated region, which actually is not that easy to say which of these variants are causal. For two reasons, this problem is hard. One is the LD becomes denser in this point. And if you look at a lot of variants here, the statistics seems very closely related. So it's very hard to just say which SNP is causal in this problem. So it's not that intuitive for some of the cases. So fine mapping is generally defined as, given the statistic from a GWAS result, can we detect which variant is actually causal? And this problem becomes hard for two main reasons. One is linkage disequilibrium, means variants are not independent. And when variants are not independent, makes the problem hard for two reasons. One is that you cannot break the region to two simpler regions and solve them independently. So it makes it hard computational-wise. And another reason that LD makes this problem hard is that if one of the variants is causal and another variant is in LD with that variance, you will see the effect on that variance. So it's very hard when you see an effect to figure out if this effect is for the SNP itself or is tagging the effect of another SNP. And another reason that this problem becomes complicated is the fact that there exists allelic heterogeneity. Or in other words, there exists multiple causal variants in a loci. And this is actually a main assumption that most existing methods try to ignore. Now, I will explain 3 methods that try to perform fine mapping, and we compare our method against them. The first 2 methods I will explain in the following slide, and the last method I will explain it in the context of our method, which is much easier. So the first 2 methods I will explain is a top K SNP and a conditional method. And the last method, we will call it one post, which is actually a method proposed by Muller et al. So in the context of fine mapping, I will try to answer 3 questions. So fine mapping is that given the associated variance, can we figure out the causal variance? So the first question you may ask is that, well, which of these variants are actually causal? And the second question you may ask is that, well, maybe we are more interested to find which gene is causal. So you want to perform fine mapping in gene level. And then I will talk about one of the applications of this is that, is it possible that we find a variant that is causal in 2 different studies? And more importantly, an example of this is the same variance is causal in a GWAS and eQTL. And I will explain why this is very important later on. So the top K method, which actually is one of the methods that perform fine mapping, is a very simple method. It picks the K most significant SNP based on the significance scores. So here, when K is equal to 5, you pick the 5 most significant SNP that are shown by the black arrow in this example. Now, this is the complicated region I mentioned in my beginning of the slide. So if you look at the real answer, the red triangle represents the true causal variance that we implanted in this region. So obviously, when K was equal to 5, the top K failed to find any of these causal variants. And the reason is that it ignores the LD. No matter what LD we put here, top K will pick the same answer. And picking the right value of K is complicated, unless you know the answer. Now, another method that people propose is the conditional method, which is an iterative method. In each step, you pick the most significant SNP as causal, and then recompute the marginal statistics of all the other SNPs, given that this SNP is causal. And you perform this iteration till all the SNPs falls below a significant threshold. So here in this example, we pick the most significant SNP, which is shown by this red arrow, and you remove its effect from that SNP and all the other SNPs. So that would drop to 0, and then you pick the highest SNP in this step, and you keep doing this. So you can see if you do the conditional twice, again you fail to find the true causal variance that we implanted in this region. And the reason both top K and the conditional method fail to pick the right causal variance is that they assume the top SNP or the peak SNP is the causal variance. And in our study, we realized actually this is not necessarily true, especially when the LD becomes complicated. So this motivated us to come up with our method called CAVIAR, which is causal variant identification in associated region, which is based on a concept called row causal set. So row causal set is a set that can capture all the causal variants with probability at this row. And in practice, we'll set this value of rho to be a very high value, like 90 or 95%. And our goal is to find all the causal variants with probability at least 90 or 95%. Now, I have to mention, this doesn't mean that all the variants that are in the row causal set are causal. What it means, if you go and validate all these variants in the row causal set, you can capture all the causal variants with probability at this row. So a trivial solution to this problem is to pick all the variants as causal set. So if you pick everything with probability 100%, if there is a causal variance, you would detect it. Now, obviously, this is not a right answer, and it's not desirable answer. So among all the row causal set, the set which has the minimum size, which means that it has the minimum number of SNPs, is the most informative one. And this has a good intuition that it will minimize the validation cost to detect actually the true causal variance. So here I'm showing the output of CAVIAR for a 95% causal set when we run it on the complicated region. So you can see actually the causal variance, the row causal set that CAVIAR picked consists of this 7 SNP and can capture the true causal variance that we implanted in this region. So at least this example shows that CAVIAR works better than top K and the conditional method for this complicated region. So how does CAVIAR work? So in order to find this row causal set, CAVIAR uses a concept called causal statuses. So for a single SNP, we have 2 possible causal statuses. Either the SNP has an effect towards the phenotype, so the SNP is causal, we represent it with 1. And if the SNP has no effect towards the phenotype, means that it's not causal, we use as 0. So if we have m SNP in a region, there are 2 to the m possible causal statuses for that loci. Now, given that, and given that we can compute the likelihood for each of these causal statuses, I won't describe how to compute that likelihood during my talk, but if you're interested, I can explain to you guys in offline. Now, we define another concept called causal set. So causal set is a subset of the SNPs in a region. And let's say for now the size of this causal set is size k SNPs. Then we can define that there are 2 to the k possible causal statuses for this causal set. Given these 2 definitions, then we can ask a question that given a region with m SNP and a causal set with size k, what's the posterior probability that this set can capture all the causal variants in the region? So in order to compute that, you can approximate it by summing over the likelihood of all the causal statuses in this causal set, which are 2 to the k possible term. And you divide it by the sum of the likelihood of all the possible causal statuses in that region, which there are 2 to the m. Now, if this ratio that you compute is greater than the ratio you wanted, like rho, like for example, if we set it to 95%, then this causal set is a rho causal set. Now, I deleted a lot of detail for simplicity of the talk and the time, but I'm more happy to discuss to you later. So we want to compare with the existing method. I explained 2 of the existing methods in the beginning of the talk. One is the top k and the conditional method. The last method is actually Muller et al. Method, which we refer it as one post. And this method tries to find a confidence set, a 95 confidence set. So it seems that it's very similar to what we are trying to find, with one big difference that they make an assumption that there is only one causal variance in a region. And this makes the problem much easier, computationally wise. However, you may have like some biases and some suboptimal answer for some region that actually exists more than one causal variance. So we compare it with this tree method, and we generate simulated data, and we implanted 2 or 3 causal variants. I'm just showing these 2 results to you guys. There are more results in the paper. And what we did is that we allow each method to pick certain number of causal variants. So we allow each method to pick, like for example, 2 causal variants, and we compute the recall rate. So the x-axis is the number of SNP that we allow for each method to pick, and the y-axis is the recall rate. So you can see in each time that we run the method, CAVIAR tends to have a higher recall rate compared to the existing method. And what this means is that CAVIAR will rank the causal variants better than the existing method. So with this, I want to go and summarize the first part of my talk, which I explained about CAVIAR, which is our method to perform fine mapping on the variance level. And this work was actually published in Genetics with collaboration with Bogdan Pasaniuc at UCLA. So the second question, the second part of my talk is that can we perform fine mapping at the gene level? And this motivated us to design a new method called CAVIAR gene. So you may ask why we did this. So the motivation is that we want to run our method of fine mapping on model organisms, especially like mouse. However, model organisms have 2 challenges. One, the stretch of LD is very large. So when this LD becomes very large, if one variant is causal, a lot of other variants are in high LD with that variant, so they seem to be significant. So it's kind of a very impractical to perform fine mapping on variance level for model organisms. And the second challenge is that model organisms have high amount of population structure, which means that the individuals are genetically similar or related. So this motivated us to come up with a new way of solving this problem for model organism. And what we observe is actually when we see a peak of association, there exists a lot of genes that harbor beneath that significant peak. So we said maybe it's possible that we can group the variants based on the genes they are. So here I'm just showing a very toy example that we have 35 SNPs, and instead of finding which variant is causal, we would try to group them, and we say, for example, gene 2 is causal. And this will speed up the whole process, as the number of genes in a genome is an order of magnitude smaller than the number of variants. So I don't go to the detail of how we did this or how we solved the problem, the problem of the population structure. What I want to show is that how when we run this program on a real data. So we have a HDL GWAS for 3 different mouse data sets. The F2 cross, which is shown in the blue in this diagram, the HMDP, the red, and the green is the outbred stock. And they are all for the HDL phenotype. And all of these mouse data sets have their own benefit. Some of them have higher resolution of fine mapping because there is higher recombination. So the LD, there was like the region was recombinate more and break more, so we can perform better fine mapping for them. So we got these 3 data set, and I'm just showing the chromosome 1 Manhattan plot for this region, and you can see there is a huge peak in the end of this chromosome. And what people have figured out is that APA2 is actually a causal gene for this phenotype. So we want to see how our method performed. So we took this 3 data set and we ran CAVIAR. And we treated HMDP slightly different, as HMDP was designed at UCLA, so we have a lot of prior knowledge at UCLA about this data set. So we asked our colleague to filter out some of the genes that they know it's not causal. So I refer to this data set that was filtered out as HMDP-CG, and we run our CAVIAR gene on this data set. And you can see there are 5 genes that is common between all of these runs. And these 5 genes are listed here, and one of them is actually APA2 that we know it's a causal one. And the other genes are actually very close in a near vicinity of that causal gene. And this work actually was published in ISMB and Bioinformatics, and we got the best paper there. So I will go to the third part of my talk, which is an application of CAVIAR model. And we want to know if a variant is causal in 2 different studies. For example, if the variant is causal in both eQTL and GWAS. Now, you may consider that why we want to do that, as most of us know that most GWAS risk loci fall in non-coding region. So it's not clear how these GWAS variants are going to affect the phenotype if they are in non-coding region. So the main hypothesis is that this GWAS risk loci affects the phenotype toward affecting the gene expression of a gene. So what we want to know is that can we compute this probability? Can we compute the probability of the same variant being causal in a GWAS and a eQTL study? And we refer to this probability as CLPP, as colocalization posterior probability. Now here I'm just showing a simple example. You can see the LD is very sparse, and I want to compute the CLPP for this variant. That the score of the significance score is very high for both eQTL and GWAS. So we kind of have a high confidence that this variant should be both causal in the GWAS and eQTL. So we expect the CLPP score to be high. Now, what can we do this with the score? If we can compute this CLPP score for all the gene in a study and for all the tissues, then we can look at which gene and which tissue is actually important with this GWAS variant. And what we can say is that we can understand a little bit of the biological mechanism of this GWAS hit. So we can say in this toy example, maybe this GWAS hit is affecting the trait towards liver and blood towards gene 4. But most probably, pancreas will have no effect for this GWAS set. So I won't go to the detail of how this method works. What I want to show is that we compare it to the existing method. One is COLOC and RTC, and we compare it to the case that we implanted 2 causal variants in the region. And I'm showing the accuracy, precision, and recall rate of our method compared to the existing method. And you can see our method have a higher accuracy and precision compared to the existing method. And I'm not showing the recall rate, as all the 3 methods tend to have a very high and similar recall rate. So what we want to do is that we want to run our method on a real data set. I have to mention one of the benefits of CAVIAR is that eCAVIAR actually is a new method that can categorize a variant into 3 categories. The variant that do colocalize, and we have a high probability that they colocalize. Variant that do not colocalize, and we have a high probability that they won't colocalize. And variants that are ambiguous at this point, so we can't make a right judgment at this point. So what we did is that we used MAGIC dataset, which is actually a meta-analysis of insulin and glucose-related trait, and we used GTEx data, which is a multi-tissue eQTL study. And we picked the fasting glucose, which has 20 significant GWAS loci that was previously reported, and we want to see if we can colocalize it using the GTEx data. But we realized actually only 3 of these variants we were able to colocalize, and the rest we couldn't find colocalized. And a majority of these variants, actually, we had a high evidence that they don't colocalize, which is actually a very striking result. And this work is actually under review, and again, is a collaboration with Bogdan Pasaniuc and Jae-Hoon Soo at UCLA and our friend at in Broad Institute. So at this point, I will go to the last part of my talk, which is the phenotype imputation. So up to now, I have mentioned that GWAS is a success and has done an amazing job to find all the GWAS associated variants that we are interested. However, the power of GWAS depends on different factors, and the power of GWAS is defined as the probability of finding a causal variance that truly exists. And this power depends on different factors. One of it is the number of individuals that you collect in a study. So if you have a lot of individuals, you have a lot of power to detect if a variant is causal. So for phenotypes that are easy to collect, like for example, HTL, LDL, or weight, you have a lot of individuals. So you expect to have a lot of power, so you expect to find the causal variants if there exists in the region. However, for phenotypes that are hard to collect, for example, the brain expression or like iron level, these are very hard to collect. Not many people want to donate their part of the brain so you can perform eQTL study to find something. So the question is that can we increase the power for these phenotypes that are actually hard to collect? Now, this problem actually kind of is similar to what people are doing at this point, and it's called intermediate phenotype in order to solve it. So in order to perform GWAS on a phenotype that is hard, what they do is that they find another phenotype that is like highly correlated to this hard to collect phenotype. And instead of performing GWAS on this hard to collect phenotype, they've performed GWAS on this intermediate phenotype, which is actually easy to collect. So an example is that instead of doing GWAS on a heart disease, what people actually do now, they collect HTL as a good approximation of the heart disease, and they perform GWAS on that, and they use the result of the HTL as approximation for the heart disease. Now, another problem that is actually related to the phenotype that is hard to collect is a missing value problem. So missing value problem is that when you collect multiple phenotypes, there are always some individual for some phenotype that you didn't collect and they are missing. So people develop a lot of methods to fill these missing values. So what we realized actually, maybe we can mix these two problems of the missing values and intermediate phenotype and solve this power issue in the phenotype that are hard to collect. So what we want to do is that we want to impute or predict this phenotype that is hard using other phenotype that we collected for that individual. And then what we do is that we perform GWAS on the imputed phenotype. So here, let me show an example. Here, we have two studies. In the first study, we have a very few individuals that we collected the four phenotypes for all the individuals in these studies. And the first phenotype actually is hard to collect. Now, in the second study, we have a lot of individuals, and the first phenotype, we were not able to collect it. So what we can do is that we can use the first study to learn the pairwise correlation between each pair of phenotype, and then using this matrix that we learned, we can maybe fill this first phenotype. And then we can perform GWAS on this first phenotype. Now, I won't go to the detail of the math of the paper. Actually, it's very interesting. I will just show one result that actually is very nice. And what we can prove is that actually the power in the imputed study with any individual is equivalent of performing a GWAS on the phenotype that is hard to collect with this many individuals, which is Rm squared and then total, and n, which is the number of individuals that you perform in the imputation study. So Rm is actually the correlation between the phenotype that you imputed and the true phenotype that you didn't collect. So let me explain this in a context of example. Let's say we want to impute the brain expression using the blood. And the correlation between, for the accuracy of our method, which actually we can represent as a correlation, is actually 0.5. So the power of performing imputation on the brain using the blood for 8,000 individuals is equivalent of the power of collecting the brain expression for 2,000 individuals. This is actually a very nice result, although there are certain assumptions that should hold for this proof to be right. But we show in practice, majority of the time for this condition holds. So actually, we are very happy with this result. And this paper was just published in the American Journal of Human Genetics. So at this point, I talked 3 of the projects that I mentioned in the beginning of the talk. However, towards the end, I will mention 2 of the projects that I did in the beginning of my PhD, which I'm actually very happy about them. The first one is actually genome privacy. So genome privacy is that individuals want to somehow share some part of their genome in a way that individual can figure out if they are related with another individual while the genome privacy is preserved. So here we know that 2 of the individuals are related and 1 individual is unrelated to them. So what they want to do is that all these individuals will want to put an encoded version of their DNA online, and the other individual will download this encoded DNA sequence and can compare with their own DNA sequence and figure out if they are related or not. Now, the way we try to solve it, actually we use the idea that used in all the devices that use the fingerprint encryption. So For example, let's say when you're trying to set your iPhone, you use your fingerprint, and it keeps an encoded version of your fingerprint in the device. Now, the next time when you want to unlock your phone, you use your fingerprint. Now, if the fingerprint is actually a close match of the original fingerprint, the phone will get unlocked. And if it's not, the phone will stay locked. And the reason I use the word close enough is that fingerprint process is not perfect. Due to the errors in the device, you will have a different fingerprint. You can't always have exactly the same fingerprint, even for the same individual. However, as long as they are close match, the phone will get unlocked. And if it's like far, the phone will stay locked. And this is actually very similar to our problem. If two individuals are genetically close, then the device will realize that these two are in close relatives, so the phone will get unlocked. So our algorithm should detect that they're relative. And if they are far relative, we would detect that they are not related. So the phone will stay locked in this example, and in our case, we will realize they are unrelated. So actually, the first paper was published in Genome Research that we show that using common variants, we can perform this privacy and detect which individuals are related genetically. And then we extend this idea to rare variants, and we show that actually we can detect further relatives using rare variants. And this work was actually published in ISMB 2 years ago. Now, I will move to the last project that I want to present. It's called pool sequencing. So I mentioned in the beginning of my talk that there exists this machine that can sequence individual and actually reduce the cost a lot. However, what I have to mention is that there are different steps in sequencing. The first step is that to extract the DNA and perform a library preparation. And after you generate the library, you give it to the machine that generates all these reads. And actually, this step that you have the library and you give it to the machine is actually very cost effective and actually is automated. But the first part that you generate a library preparation is actually very expensive and labor intensive. So the question is that can we pool this individual together in order to reduce the sample preparation cost? And people have done it, and we want to ask some interesting question in this context of pooling. So the first question we were interested to ask is that do all samples contribute equally to the read that is generated from that pool? Which means that do all individuals have the same abundance? And actually the answer to the question is no. Some individual will contribute more and some individual will contribute less. And this work actually was published in Recon, which we have a nice likelihood model to find the right abundance for each of the individuals. Now I explained the method in the context of pooling, but actually you can see that this is very similar to the problem of bacterial reconstruction, which you sequence a group of bacteria and you want to know which set of bacteria exists and what's the abundance of each of the bacteria. And actually we show in the paper that we can apply it to that data set, and we got really good results. And the last question that we were interested in the context of pooling, can we perform a dense genotyping using pool sequencing? And this work was actually published in a Cellmore conference in 2012, where we use a likelihood model and a compressed sensing to solve this problem, and both methods tend to give very good answer and actually very close. So at this point, I mentioned some of the projects that I did during my PhD. I want to acknowledge the people who were involved in this project. There are too many people to acknowledge, so hopefully I didn't miss anybody. So I want to acknowledge the people in our lab. I want to acknowledge the Jake Luce's lab. They had a huge contribution and giving the data to us. I have wrote a lot of papers with their lab, and I learned a lot. They worked very hard, especially Jake himself, that of course he's not here today. He had to go to Europe, but I used to get email from him at 6 a.m. in the morning. So I want to thank the crypto group at UCLA. They help us with the crypto paper. I want to thank Jason Kong Lab, which they're helping us to speed up the CAVIAR method. And a lot of other people from UCSF, Bogdan Lab here at UCLA, and Aaron, which we have wrote 2 papers together. And a lot of other people that I mentioned here. I want to thank some of my friends and family, which made my PhD life much easier. And some of them are at UCLA, some of them are UCSF, and some of them are now working. I want to mention one person that actually I didn't put here is Wang. I know he wanted to come to my defense, and he mentioned that I should put it on Friday, but there were like a lot of complications that I couldn't put it. So he really wanted to come to the defense. So I'm very happy that he's such a good friend. And I want to thank my parents that actually they live in Minnesota. In the end, I want to thank the committee member. I want to thank Jason and Wei, although I never worked with them directly. But, well, I will get to that. Well, Jason, I know his research because every conference that I go, everybody will talk about Chrome HMM. So, and Wei, actually, I know his work, her work, actually before I started my PhD about the graph mining. What I have to say is actually, me and Jason have paper together. Actually, two papers. One is that eGene paper, and apparently there is another paper that is under review from Dan Gashfield's lab. And more importantly, the reason I know their research is that I was roommate with Chelsea and Pete for 2 years. So we used to talk about research, what we do in the lab. The person I want to thank is Nelson. We are working on different projects, especially an eQTL study with Nelson. But more importantly, what I want to thank Nelson is that he gave me an NIH fellowship in neuroscience, which I didn't have to do TA because I already have done 7 quarters of TA, so that was really great. And then Nelson is the head of Depression Grand Challenge at UCLA, that they are planning to sequence 100,000 of individuals who have depression at UCLA. So it's a great project, and it's a huge project. And as part of this, Nelson had the idea that maybe we can come up with an app that can monitor this individual who have depression. So he asked some of the people, me and 3 other people, to take this course at UCSF to learn how to make a startup. And the course was that in the course of 7 weeks, you have to interview 70 people unrelated to you about your idea in the startup. And it's actually very hard to find 10 people that you don't know to interview. And I remember one of the requirements of the course written in bold that if you're not used to your teacher criticize you in front of other people, you should never take this course. And I was like very saying why they did that. This doesn't make sense till I took the course and I realized why they say that. And in the end, I want to thank Bogdan. Although Bogdan is my committee, but he acted as a second supervisor. In my last 2 years, all my papers are with him, and his knowledge in GWAS is amazing. He has read all the papers, and he always can send me comments that, why didn't you compare with this method? There is this method, and if we don't compare, the reviewer will ask it. And majority of the time, if I ignore it, the reviewer will ask for it. So I'm not sure. Maybe he was one of the reviewers as well. But so I want to thank 4 people that had a huge effect before my PhD. One of them is actually my brother, who's actually faculty at UC Davis. And the good thing is that he was the person who first introduced me to the field of computational biology. Although now he always makes fun of the problem I work. He says, I'm not sure if GWAS will solve the problem, but it's actually very interesting to do brainstorm with him. The second person I want to thank is my master supervisor in Simon Fraser, which I learned a lot. He had a theory background, and he taught me how to use a computational problem and use the technical computational techniques that we learn in algorithmic to solve this problem. And then I moved to University of Washington, which I worked with Evan Eichler, and at that time, John Alcon was a postdoc in that lab. And I learned a lot of sequencing at that time, and that was actually very important for me because when I joined Czar lab, Czar had a huge confidence in me that I know sequencing. And he gave me a lot of cool projects, and I'm very grateful for that. And then, of course, I want to thank Eleazar. I know I should say a lot of good stuff about Eleazar, and all of them are true. But one thing I want to say is that Eleazar is very patient, and that was very nice during my PhD, so I wasn't scared of Eleazar. The second important thing is that Czar sends everybody to all the conferences. That's a very important thing. I don't think so that many faculties do that. And that gives a very good chance for people to go and get exposure to other research in different conferences. And Czar, I remember he sent all of us to China for recom, which most of us didn't have paper. Like I think 2 or 3 of us had, but 7 people in the lab was there. And I can say that I will never email anyone in my life more than I email Czar. That's for sure. And I'm sure I will never get response from anyone more than Czar. And I'm not sure if Czar will get upset, but I was trying to figure out when Czar sleeps. So when I would send emails, I would get response back from Czar in less than half an hour. And I used to send email like 1 a.m., 2 a.m., 4 a.m., and then like 9 a.m., like in the middle of afternoon, 8 p.m., and still Czar will respond. So I was very surprised that when does Czar sleeps. So I'm still confused about that. But I'm very happy that Czar will respond to any email I would send, like simple emails that, hey, Czar, I don't have a laser pointer. Can you bring it to my defense? Or, Zar, what's going on with this? Zar, what's going on with this paper? Like, I'm very stressed. And he said, don't be stressed. I know a way to fix it. So I'm very happy to work with Zar. And actually, it was great. The 6 year was great. Thank you, Zar. And I'm ready to get the questions. Thank you.
Okay, so traditionally, we start with questions from the committee. Maybe we can start with Nelson, since he's on the phone. Nelson's there?
Nelson?
Yeah.
Hello?
Okay, or maybe while he's setting up, maybe on the committee.
Yeah, hello?
Oh, yeah.
Hey, Nelson.
Yeah. Any questions from Nelson?
Okay, yeah, so I mean, obviously that was a tour de force and really incredible. So, Farhad, I just have one question which relates to the phenotype imputation, which I think has incredible promise. But one of the things that I see is, you presented a very simple case where the correlational structure was pretty straightforward. But I think that for a lot of the really practical problems for phenotype imputation, you're going to be dealing with things where it isn't going to be so easy to predict the correlational structure. And I just wonder if you have any thoughts about how you would deal with that.
So actually, the correlation structure is actually, here we show correlation structure. And more importantly, this should be the genetic correlation between the phenotype. I agree, some of the phenotype, if you don't have enough individual, is going to be hard to collect. And we won't have enough accuracy. But the good thing is that even if you have low, like if your prediction of the correlation structure is not very good, we showed that still the false negative rate is controlled. So if you find something, you can be confident that it's actually right, but you may miss. So there are some phenotypes that actually they're very problematic, and we don't have good correlation for them. So as soon as we get good phenotypes that are correlated with the phenotype that is hard to collect, this method should work well, but it's not perfect at this point, for sure.
Yeah. Okay. Thanks.
So I guess two related questions about the CAVIAR. So you're making this assumption that, or you're motivated by there being multiple causes.
Yes.
In the case where there's only one causal SNP, so the comparison you showed had 2 and 3. How
Yes.
does it compare when there's only
Yeah, actually, that's a
one?
very good question. And we showed that the result would be very similar to the case of the case with the result of the method that they use one causal variance. And the reason is that they are trying to solve the same problem. And most probably, the reason that's happening is that the likelihood of the causal statuses that SNP is causal will be so high that it will kind of ignore everything else. So.
And then, sort of related question. I mean, one could also think of a model where every SNP is contributing some effect.
Yes.
That it's not like this. You're either causal or not. So then, what would your method sort of how it would behave in that type of architecture? And then, to what extent do you think it's really the case that lots of loci are modeled by 2 or 3 and not just 1, or everything has some tweak?
So actually, that's a very good question. So I have to mention that when we are modeling, if there is 2 causal variants, we assume they're coming from a specific distribution, the effect size. So I didn't show that, okay? But I completely agree. If you have a region that all the loci have weak effect, then this model will have problem. Although you can put the computational costs and you try to go and enumerate and try to figure them out. So there's this bound that we put it, for example, 6 causal variants. So if you have more than 6 causal variants, then the result will be a little bit off. But it won't be that it won't pick the wrong SNP. It will pick more SNP. So your set will get really big. So if you have a region that all of the SNPs are causal, but they are not in LD, they're not all in perfect LD, actually the method will pick all of them. Because all of the likelihood of these cases will be very high. So in the end, the method has to pick all of them to reach the 95% probability. Is that clear?
But what's your reason for or do you have any evidence to believe that there's lots of locations that are sort of well modeled by 2 or 3?
Well, there are some biology paper that they show that there is more than one cause. Actually, we have a paper that shows in eQTL study there is a prevalence of allelic heterogeneity in eQTL studies. And eQTL is actually one of the studies that people believe that there shouldn't be more than 2 causal, but now we see that 20% of the genes have 2 signals, 2 independent signals. So definitely assuming that there is only 1 causal variance is not a good thing to do at this point.
Okay.
I'm not sure if I answered the question right. So
yeah, I mean, but then those that say that there's more than one, then the question is
how many there are?
yeah.
So actually, that's very hard. We have a method that says how many there are, but it has a limitation that it can tell only up to 6. After that, it will give a very high probability that is more than 6. So you can run that first and have an intuition what's going on in that region.
And for the sort of gene extension method, when you were doing CAVIAR with the gene level,
Yes.
How were you associating SNPs with genes?
Oh, we did something very simple. We just look at the distance. So we group a variant based on how close is it to which gene. I know it's not a good idea, but the method can incorporate any distant matrix or like any grouping you will give to it. But what we use in the paper was just based on distance. What you can do is that based on the eQTL expression that they are actually affecting. But at that point, we just use the distance to solve the problem.
Should I keep asking?
You can.
One last one.
Okay.
You're allowed.
So when you're doing the, so part of this was motivated by you wanted to, with the validation for the CAVIAR, like your metric was you wanted to have sort of the smallest number to validate. But from the point of view of if you had multiple different loci in the genome giving GWAS signal, would you be best off sort of just taking like the top hit from each loci, or would you trust your like second hit from your CAVIAR output? If somebody had like a budget to do like 5 validations, would you recommend that they take the top signal from 5 different hits, or would they put 5 validations on
One loci. Well, that's actually if they are in independent LD, I would say to run it like CAVIAR on each one independently. It will give like it's better because you know that, for example, this region has its own causal variance. But the assumption that you have to pick the pick one is actually usually it's not a good assumption. So I would say to run CAVIAR on that region independently. It's very high probability it will pick the pick one, but it's possible that another SNP will be picked. Like for example, in the case that I showed 2 causal variants, the middle variance was actually had a higher score, but CAVIAR will first pick these 2 and then will pick the top one.
I mean, I guess sort of the question is CAVIAR, I mean, you're sort of saying it's giving you a set within one independent location, but then if somebody's sort of trying to decide what to validate and they have multiple independent loci, does it like rank among the different locations?
It will rank, actually if you give the whole region, it will pick variants independently as well. So it won't miss anything if it's really causal, but it may pick a lot. So it's better that you separate them. Oh, we ranged. You mean what kind of effect size we use?
What kind, not the size, but maybe there's some different type of effect. Especially you talk about more than multiple effects, but not just a single effect.
So what we did is that we used the power. So we tried to put the power of detecting of loci to be like 50%, and then we simulate data based on that. So based on the number of individuals and the effect size, we will simulate NCP, which corresponds to the effect size, and then we see how the model works. Now the question is that if you're asking if we put epistasis effect, that 2 SNP together make effect, no, the model make an assumption that all the effects are like not epistasis, they are like.
Right, but you simulated, you test that if there is such effect.
We didn't test the epistasis effect.
You know, how sensitive the model is.
We didn't test the epistasis, and I think epistasis will have its own model, and we have to correct for that actually, because I think the statistics have a specific part, and I think we will miss the epistasis effect. But if there are 2 independent signal, we will detect it. But if their signal is the effect is together in epistasis format, we may not find it actually.
Right, but in reality, typically it's not. You know, there's some, right?
No, if they're in LD, it's okay. If
No,
they're,
not in LD.
yeah, then we will,
Some-
yeah,
I mean, but,
yeah.
you know, do you, because if you miss that completely, or you detect some,
I didn't do experiments, so I don't know that.
yeah, I would just say how sensitive the model.
I don't know. That I have to do experiment. I don't know.
My turn? So I have a question on the phenotype imputation. Have
Okay.
you tried, so let's say that you measure a lot of phenotypes and you don't have any missing ones. Is
Okay.
it better to just re-impute each of them at a time and then associate that to the trace? Could you be, let's say you have 10 phenotypes, all measured in all individuals, then now you can associate phenotype 1 to, or you can associate SNPs to phenotype 1, and now you can also associate SNPs to the imputed version of phenotype 1 from the rest.
So I didn't do that, but I think Nancy Cox's paper of imputing the gene expression, and they show that actually it's better to use the imputed version because it will remove a little bit of the noise that they observe. But I'm not 100% sure. But I think it.
Right, but I think it's slightly different because they're imputing from genetics, right? And you're imputing from other phenotypes, as far as I understand.
Well, here's the thing. What we impute, we rely a lot on the genetic correlation. So if what we are imputing is actually the genetic similarity. So if the genetic if two phenotypes are not genetically correlated, actually the method will not find it. So it makes an assumption that this effect exists genetically in this variance have effect in both of the phenotype in order to make sure it will detect it. So it may improve the result because it will remove some of the noise that we collected in data, but I have to test it to make sure. Yeah.
Right, and the second question or a follow-up to that is, why do you only look at the genetic? Why can't you impute the phenotype from the other if they share some environmental factor?
Well, that proof was harder, so I can. The proof of genetic was much easier. Actually, it wasn't easy; it was really hard. So we have to bring another person to complete it. But the environment is. I'm sure it should work in practice, but I'm not sure the proof that we said about the power will hold, like it will go true. But definitely it will work, and I think it will give better result. But I'm not sure then we can talk about the power, the formal power that we have, that we can prove something about the power, what happens. That, I don't know if it's that easy to prove. Because that will have its own correlation structure, environment has its own correlation structure, and then it will be a little bit messy for the math.
Sure.
But it should work, but
Maybe this is something, but isn't like the power just simply, the accuracy? So in terms of Pearson, so whatever the accuracy is from whatever predictor you have in terms of R-squared, the power would nothing be than R-squared times the sample size. So if I give a predictor that
Yes.
gives you higher
Yes.
R-squared,
yes.
then
No, your power will get increased as well because your imputation is better, but I'm not sure if we can prove that the power is exactly the equivalent of this formula. It may be a little bit different formula, but for sure the power get increased because you're doing a better job.
I see. In some sense, what I understood from your math is that will be a lower bound. In real life, you might actually use much better than that.
Yes.
R-squared.
Yes.
And,
Yes.
because, you can leverage other types of correlations
Yes.
between the
Yeah, that for sure.
research.
That's for sure.